tricky. In the cytokine column, you can see that this data set involved the treatment of IL-2 and IL-15. However, they don't explicitly tell you which condition is a cytokine treatment and which condition is a control treatment. And that information is actually available here through the W withdraw or activated. They activate the cell with different cytokine first, and then withdraw the cytokine a little bit to remove the cytokine effect. So in this case, actually the A will be the treatment condition, the W will be the control condition. So we need to put the treatment information here. Okay. However, we cannot leave W or A here because these things are not our control vocabulary. For A, we need to put them either as L2 or the L15. For W, we need to put them as the control. So what we do is to join the cytokine names on after the treatment W or A symbols. We do a join back, okay? And then we are going to replace. So for example, for anything like withdraw, and then a cytokine, we need to replace them with control condition. And here, we can use some regular expression to make this more automatic. This pattern means we are matching something like W dot IL and then some numbers as the interleukin names. After this replacement, you can see all of these things become the control. And for A, we need to replace the A.15 IL-15 into just IL-15, and the replacement should be easy. Just simply remove the A dot with empty string. Right now, the control is ready. And then we need to look at what is the cell model, which is the condition. Cell type. Then we just copy them here. However, here is a tricky part. The control conditions are actually very different across different rows here. For example, among the IL-2 treatment, the control conditions are actually these rows. And among the IL-15 treatment, the control conditions are actually these rows because you treat them with one specific interleukin first and then do the withdrawal. So these control conditions are IL-2 or IL-15 specific. What we do is just simply put the cytokine information after the condition, after the cell model. So in this case, when we analyze them, we will just look at each group first and then look at the treatment conditions within each group. This experiment also has some donor information, donor 1, donor 2, donor 3A, 3B, or 4. What we could do is put the donor information into the subcondition. And they also have some different protocols of RNA extraction. You can put this information either into the condition or maybe into the subconditions. So for example, we can put the RNA extraction approach into the subconditions. Now this annotation is ready and you can submit.